Hello, a good day to all. I'm Christine Chung, an assistant professor from Nanyang Technological University, Singapore. Today, I'm honored to introduce our laureate for the next plenary lecture, and he's Professor Thomas Südhof from Stanford University. He's best known for his discovery of the machinery regulating synaptic transmission. In the 1990s, he has discovered the integral protein components that are critical to the fusion process of vesicles and the presynaptic terminal's outer membrane, a process which allows the vesicles to spill its contents, which are the neurotransmitter molecules, as a mean of how neurons communicate, by far the best known and understood phenomena in neuroscience. For this seminal contribution, he was recognized with the Lasker Award in 2013 and in the same year, a Nobel Prize. Beyond research, Prof. Südhof has worked with multiple pharmaceutical companies, has co-founded 4 biotech companies, and served on the board of directors and advisory boards of multiple biomedical firms. Today, we look forward to his exciting lecture on understanding synapse formation in neuropsychiatric and neurodegenerative diseases, a multifaceted challenge. Prof. Südhof, over to you.
Thank you. So let me put up my slides here. I hope you can all see it. It's truly a pleasure for me to talk to you here today. I decided in thinking about what I would talk about, not to give one of the sort of overview talks that I sometimes do. I was also deciding whether I should talk about industry and the challenges of approaching treatments for neuropsychiatric and neurodegenerative diseases. I decided not to do that either. I decided, and I hope I'm right, it's too late now, to instead talk about actually the scientific challenge that interests me most at present, and that is directly related to diseases, but hasn't been able yet to solve these diseases. Before I start discussing this with you, I would like to just briefly tell you that I am involved in multiple consulting activities besides being an employee of the Howard Hughes Medical Institute and Stanford University. These are listed here. I won't go into any of the details because nothing I'm going to talk about today has anything to do with my consulting. My academic work focuses on the brain, of course, but my consulting is actually much broader than that because I am interested generally in the challenge of conquering disease. So what I want to do today is first tell you about the problem that we are trying to solve now, that is the most interesting question to me, which is synapse formation. And I want to tell you why it is important. And then I want to illustrate the approach with one particular example that I think most easily illustrates what I'm trying to tell you. So let me start at the beginning. What you see here is a human brain, dead brain, unfortunately, to illustrate how huge this organ is. It's enormous. It contains 10 to the 12 neurons at least. Each of the neurons is connected to other neurons by specialized contact zones called synapses. Thousands and thousands of synapses. Probably an underestimate what's shown here. The complexity that this creates is vastly higher than, for example, the complexity of the human genome, suggesting that the complexity of how the brain works is likely to be higher than that of the genome. And as you all know, we don't understand how the genome works. So we have some work cut out for us. Neuroscientists study the brain using a vast number of approaches. They often argue, they sometimes even fight, but there's a common concept that underlies most, if not all, of current neuroscience. And that is the concept of the neural circuit. Neural circuits connect neurons into networks that process information, and these connections are formed by synapses. What you see here on top is an electron micrograph of a classical synapse, synaptic vesicles filled with neurotransmitters next to the actual synaptic contact. They fuse and transmit the signal. What you see here in a very simplified way is a microcircuit where the left blue excitatory neuron excites the right blue excitatory neuron and the red inhibitory neuron, creating feed-forward inhibition. Neurons form circuits, and the circuits process the information by transferring the information at synapses. During the transfer, synapses change the information. They represent the fundamental computational unit of the brain. Our lab's major objective is to identify the molecular rules, sort of the logic, if you want, that determines how synapses are formed and how they're specified. How is it that neural circuits can be built initially and can be restructured throughout life, eliminated and rebuilt, always, all over and over and over? What are the molecular underpinnings that makes this possible? And the fundamental proposition of our work is that synapses are specialized intercellular junctions that are controlled by bidirectional signaling across the synaptic cleft, as shown here with this red arrow. In other words, that the presynaptic side not only secretes neurotransmitters, but that it also, by a different mechanism, tells the postsynaptic side where to be and what properties to have. And conversely, the postsynaptic side does the same, reverse to the presynaptic side. This bidirectional signaling at synapses forms the synapses. It creates them, it maintains them. It organizes the presynaptic active zone and recruits and regulates postsynaptic receptors. So this is really, we believe, the key to understanding how circuits are built in brain. And it is not surprising that many adhesion molecules form complexes across the synaptic cleft corresponding to that red arrow, and thereby form signaling machines that perform this bidirectional signaling. What you see here in this next slide is an overview of some of the more prominent adhesion molecules that have been postulated to be candidates at the synapse. There's actually a lot. On the left side is the presynaptic side, on the right side is the postsynaptic side. You can see that especially on the presynaptic side, there are hub molecules that interact with many postsynaptic molecules. So we have a wealth of candidates here. Moreover, interestingly, many of these candidate synapse adhesion molecules were implicated in neuropsychiatric disorders and actually also in neurodegeneration. They were particularly implicated by human genetics in autism and schizophrenia. I want to illustrate this because it exemplifies the importance of understanding this question using the Neurexins, which is this hub molecule on the presynaptic side that interacts with a number of postsynaptic receptors, as you can see here. What is interesting about Neurexins is that mutations in Neurexins are still, among all the genetic causes, the single most common single gene mutation that predisposes to schizophrenia. Such mutations also predispose to autism, they predispose to Tourette's syndrome, and they predispose to other neurodevelopmental disorders, suggesting that they are key in the development and maintenance of a normally functioning brain. As it turns out, these mutations, all of them, are actually deletions because the Neurexin 1 gene is very large. And so there are many deletions that have been identified. This gene is prone to deletions because It has lots of repetitive elements. Although it's a large gene, it doesn't overlap with any other transcription unit, not a coding and not a non-coding DNA. The Neurexin 1 gene encodes only Neurexin 1 and nothing else. What's interesting also, as I mentioned already, is that there are many neuropsychiatric disorders that are associated with these mutations, suggesting that there must be some fundamental dysfunction and that possibly these phenotypes are different because of genetic background effects. To validate these mutations, to validate this as a pathogenic mechanism, we started some time ago a large multi-center project that is illustrated in this slide. First of all, we took patients' iPS cells that carried heterozygous Neurexin 1 mutations. We generated the neurons from these iPS cells, and we analyzed them functionally at multiple sites to ensure reproducibility. Second, we also tested whether the same mutation under identical conditions produced in mice, when artificially introduced into ES cells, would actually cause same phenotypes in mice and humans. So let me focus first on the patient-derived neurons. When we analyzed these neurons, we found a fundamental deficit in these human neurons derived from iPS cells in synaptic transmission. This is shown here in multiple pairs of controls with patient-derived neurons. And what you see here is that there is a decrease in synaptic strength. And that decrease in synaptic strength induced by the Neurexin mutation is associated with an increase in the coefficient of variation, suggesting that there's a decrease in synaptic strength because there's a decrease in release probability. This was further confirmed by using paired pulse measurements in the same pairs of mutant versus control neurons. And so these experiments outlined a reproducible synaptic change, a reproducible synaptic deficit that causes a change in how these neurons actually transfer information, suggesting a fundamental impairment, which is actually kind of surprising because this is a heterozygous mutation. And so because it is heterozygous, we also wondered if this might be something that is more easily observed or happens in human neurons as opposed to mouse neurons. We did this in particular because in previous studies in mice, we hadn't observed a heterozygous mutation effect. So we engineered human and mouse neurons that had an inducible mutation that was identical in every respect. We then used these ES cells, engineered stem cells from humans and from mice, and we induced these mutations. We compared their phenotypes. And I'm only going to show you one piece of data here because we have very little time together. And this piece of data demonstrates that, again, with this engineered mutation that basically replicates the mutation observed in human patients, we see this deficit, the synaptic deficit, now measured as a decrease in mini frequency, as you see here on left in the orange one. But when you look at the right samples, which are from mouse, you can see that the deficit isn't there. So indeed, there is a specific deficit that is apparently present in human but not in mouse neurons, even though they are identically prepared, the identical type of neurons, suggesting that for an unknown reason, the human environment, the human genetic environment, makes them more susceptible to a loss, a heterozygous loss of Neurexin 1 function. And this basically indicates a reason why this mutation may actually have this deleterious effect in human patients. So this is a functional phenotype. Is there a molecular correlate? Does something change in these neurons? it turns out that the synapse density doesn't change. The number of synapses doesn't change. Neither do dendrites and axons. So we were a little disappointed by this because we had hoped that that would occur. And we looked at the proteins in these samples. And I'm only going to show you one result because it's the most striking among many proteins. And in fact, mRNAs, as studied by transcriptonics, the only thing that we found changed was an increase, surprisingly, of a protein called Cas, a protein that we had identified decades ago as a cytoplasmic binding partner of Neurexins, indicating that that correlates with the loss of function of the Neurexin 1 and correlates with a functional change. And this was also replicated, as shown here in the bottom, in the engineered Neurexin 1 conditional knockout mice, suggesting that it is truly due to a loss of Neurexin 1 expression. So what I've been trying to tell you here in this small vignette, as summarized by these conclusions, that heterozygous Neurexin 1 deletions in human neurons derived from patients with schizophrenia, but not in identically generated mouse neurons, impair synaptic transmission. This impaired synaptic transmission is due at least in part to a decrease in release probability, and it's not due to a change in the synapse numbers. This is due to the effect of Neurexin, to the function of Neurexin, in specifying the properties of synapses, in this case, the release probability. And the implications for neuropsychiatric disease is that with this highly reproducible synaptic impairment, we have a direct correlate of the human clinical syndrome with a fundamental biological change. It is possible that this manifests in patients in different ways depending on genetic, developmental, or environmental covariance. But with this observation, there is now a handle on the physical nature of the Neurexin 1 deletion-induced impairment, and thus a way forward to further understanding of the system. However, this is a vignette. What I really wanted to tell you about as another example is about Latrophilins, which is another class of synaptic adhesion molecules. And I wanted to tell you about them because they turn out to be key regulators of something else, different from Neurexins, even though they have some interactions with Neurexins. They turn out to be key regulators of synapse formation. Key regulators that are also closely linked to neuropsychiatric disorders, but in this case, to a different neuropsychiatric disorder, OCD, and not to schizophrenia. So let me move on to the second part of what I was going to tell you today, which will be slightly longer than the first part, and which will tell you how latrophilins control the formation and specificity of synapses. Latrophilins belong to a family of proteins that are called adhesion GPCRs. They're called adhesion GPCRs because they're G protein coupled receptors, GPCRs, that are also adhesion molecules. What you see here in this slide is a diagram of the general design of adhesion GPCRs. They contain extracellularly large sequences, different from most other GPCRs, that include various adhesion domains that differ between different types of adhesion GPCRs. They contain a canonical set of adhesion GPCR domains, a gain domain shown here in dark green that contains the so-called GPS sequence, which mediates autoproteolysis, and a classical GPCR 7 transmembrane region domain. Finally, cytoplasmically, they contain a variable cytoplasmic region. This is the general structure of adhesion GPCRs, and they are all united by this one set of canonical domains, which are the gain domain and the 7 transmembrane regions. And the gain domain is interesting because it's found only in adhesion GPCRs and actually in some PKD-associated proteins. The gain domain mediates autoproteolysis. What it does is it's a domain that catalyzes its own proteolysis, its own cleavage. But after the cleavage, the two parts remain associated with each other. About 10 years ago, in collaboration with Axel Bringer's lab, we solved the crystal structures of several gain domains. What you see here is, in fact, the latrophilin 1 gain domain. It is composed of two subdomains, the yellow helical domain and the pink beta strand domain. The gain domain cleavage site is where the asterisk is. This asterisk and the blue finger that you see here, the blue strand, is a beta strand that remains associated with the pocket after the cleavage happens where the asterisk is. So as a result, although this gain domain mediates cleavage, it's unclear whether it actually mediates the dissociation of the two parts, or whether that cleavage for some reason is just an intrinsic component, intrinsic property of this. One plausible idea in the field of adhesion GPCRs is that the adhesion binding to the bi-adhesion domains causes a pulling off of the whole gain domain, leaving the finger, so to speak, exposed, and that finger then acts as a new free N-terminus that inserts into the pocket of the GPCR domain as a tethered agonist, called in German Stachel. That idea is very attractive because it presents a possible avenue of activation, but it may or may not be true. We are interested in latrophilins and adhesion GPCRs because, as I will tell you, they are crucial components of the adhesion machinery that makes a synapse. And in the case of latrophilins, the adhesion domains that are present in this adhesion GPCR consist of a lectin-like domain at the N-terminus and an olfactomedin-like domain immediately afterwards. As it turns out, these two domains bind to multiple transsynaptic ligands. The lectin domain binds to tenuins, which are large evolutionarily conserved cell adhesion molecules of a very unusual structure that I'll show you a little bit later. Whereas the olfactomedin domain binds to flirts, which is another evolutionarily conserved family of leucine-rich repeat domain adhesion molecules. These interactions mediate transcellular bindings between cells, demonstrating that they are truly adhesion interactions and not just cis interactions on the same membrane. Now, latrophilins were actually originally purified more than 2 decades ago as potential receptors for alpha-latrotoxin, which is a toxin that acts to trigger neurotransmitter release from presynaptic terminals and also binds to Neurexins, suggesting that they are presynaptic molecules. But this identification, this purification didn't tell us what they do. So what we embarked on some time ago is to create conditional knockout mice to study their function. And what I will tell you now is what we learned from these conditional knockout mice. When we put cultured neurons into a dish from conditional latrophilin knockout mice, and we sparsely transfected them with either a mutant Cre as a control, called delta Cre, or with Cre recombinase, we got a very surprising result. What you see here is an image of a neuron in green with all the dendrites. That's the infected, transfected neuron. You can also see other neurons, or at least the nucleus of other neurons, stained for the marker NUN. So what we do here is only 1 in 20 neurons is transfected. Most neurons are not. So we are measuring the effect of a postsynaptic cell autonomous change. When we did this, we found that the postsynaptic deletion of Latrophilin 2 caused a major decrease in the number of synapses and spines, as shown here. There was an almost 50% decrease in spine numbers, suggesting that latrophilin acts postsynaptically to control the number of synapses that are formed onto a neuron from presynaptic partners. This was a surprise because it suggested that latrophilin 2 functions postsynaptically. Moreover, although with RNAi we can see all kinds of phenotypes in terms of synapse loss, with genetic manipulations, including CRISPR, we have never seen something like this. This is more severe than anything we have seen. So this is the most severe synapse loss phenotype, suggesting this is really important. So we initiated a comprehensive analysis that is still ongoing. In the work up to now, we compared latrophilin 2 and latrophilin 3. Latrophilin 1 hasn't yet been studied. What you see here is a hippocampal section that analyzes the relative distributions of latrophilins 2 and 3. And what we found is latrophilin 2 is primarily in the distant dendroids, the stratum lacunosum moleculare, whereas latrophilin 3 is primarily in the stratum oriens and stratum radiatum. What this means is that they are differentially distributed in the same postsynaptic neurons. They go to different places. They have distinct, presumably distinct functions because they have distinct localizations. What is interesting is that these distinct localizations correspond to distinct hippocampal circuits. This is illustrated here where you can see a schematic of the hippocampus. The inputs are via the perforant path. There's both a direct and a trisynaptic perforant path shown on the right. The blue direct one forms synapses that are formed on the stratum lacunosum moleculare, where the latrophilin 2 is. The red ones, via the trisynaptic pathway over the granule cells in CA3, form Schaffer collateral synapses on the stratum oriens and stratum radiatum, where latrophilin 3 is. So the localizations of latrophilin 2 and 3 in the CA1 neuron corresponds to distinct circuits and distinct input synapses. Obviously, this implies that they function in these distinct circuits, and given the first result that I showed you, that they may determine the synapse numbers. To test this hypothesis, we performed sparse infections of CA1 neurons, as shown here. These were done at P0 at birth. Then 3 to 4 weeks later, we sacrificed the mice, we cut slices, we patched the mice, the CA1 neurons in these slices, and we filled them with biocytin to analyze their morphology. We found that there was, in the case of the latrophilin 2 deletion, a selective loss of spines and synapses only in the stratum lacunosum moleculare, but not in the stratum radiatum and stratum oriens, which makes sense because this is where latrophilin 2 normally is. To confirm the functional importance of this, we performed electrophysiology. In the same slices, we patched the CA1 neuron again, and then we stimulated for the same neuron either the inputs of the Schaffer collaterals or the inputs of the perforant path. And this is abbreviated here as you can see, as the stratum SLM, which is the inputs of the perforant path, or the stratum radiatum, which is the input of the Schaffer collaterals. When we quantify these, what we find is that the inputs of the Schaffer collaterals are actually increased in synaptic strength, presumably because there might be some kind of compensation. Whereas the inputs from the perforant path in the stratum lacunosum moleculare are significantly decreased, confirming that there's a change in synapse numbers that causes a change in overall synaptic strength of the connections. So this was for latrophilin 2. What about latrophilin 3? Does it perform a similar function in its dendritic domain, which is the stratum oriens and stratum radiatum? We use the same approach. In fact, that's the same pictures, just to make sure that people understand that this is the same approach. We again quantified spine densities and synapse densities. Now we found that there was a significant decrease in the stratum oriens and stratum radiatum, but not in the stratum lacunosum moleculare. And we confirmed this by input-output measurements of synaptic strength. We were getting better at them as we were doing them, and you can see there's a large decrease in the Schaffer collateral synapses that form on the stratum oriens and radiatum, but not in the other synapses, the perforant path synapses that form in the stratum lacunosum moleculare. So these data confirm that in the same postsynaptic neuron, latrophilin 2 and 3, dictate the number of synapses in different dendritic domains, in distinct types of synapses. They correspond to distinct circuits, but seem to be performing similar functions. And what I want to do for the remaining minutes of my talk is to tell you how latrophilins actually work. And how we did this is by rescue experiments. We made mutations that first would test different ligand binding sites as well as autoproteolysis. And this way we could test whether these activities, biochemical activities of latrophilins, are functionally important. We confirmed these mutations using cell adhesion assays that basically demonstrated that they are surface transported and selective in their biochemical changes. We then performed rescue experiments, and we found in a nutshell, as shown here, by measurements of excitatory synapse density and by measurement of the so-called mini frequency that I already told you about with the Neurexin mutations, that when we mutate the ligand binding sites, either for latentulin or for flirts, we block functional recovery. But when we mutate, as shown here in the light green bars on the right, autoproteolysis, when there's no longer autoproteolysis, rescue is fine. So autoproteolysis, at least for this signaling, is not required, whereas ligand binding is required. But surprisingly, it seems as though you need both ligand binding sites, which was a huge unexpected surprise. So we wondered if we could confirm this with real synaptic transmission experiments in vivo, using again acute slices and input-output curves. And what you see here are such measurements. I'm not going to go into the details for reasons of time. Just to summarize, as you can see here, the wild-type latrophilin 3 rescues fully, as shown here in pink, but the mutant latrophilin 3 that lack binding of either the attenuants or flirts, but maintain binding of the other one of the ligands, are unable to rescue, suggesting that both ligands must bind. Now, this was quite unexpected, sort of revolutionary, and prompted us to go further and test it with an independent method with retrograde rabies virus tracing. I'm not going to go through this method because of a time limit. Let me just tell you that you can see here in this green picture, which shows you a section through the hippocampus on both sides, that when you inject rabies virus on one side, you can visualize synaptic projections onto those cells as green cells on both sides, because both sides contribute to those cells that you injected into. And you can quantify this. So this allows mapping of bilateral inputs and allows testing of latrophilin 3 function. And what you can see here on the right is that the result is exactly the same again. The ipsilateral and contralateral Schaffer collateral inputs are severely impaired upon the Latrophilin 3 deletion, whereas the entorhinal cortex perforant path inputs are not. So that is really surprising. And it basically means that you need two ligand binding sites for this function. And it suggests that there is coincidence signaling. And let me illustrate this with the crystal structures. What you see here are in green down there, tenurin crystal structures, huge molecules. They bind to the pink latrophilin N-terminal lectin domain. You also see in light blue FLIRT3 crystal structure, which binds to the olfactomedin-like domain. In addition, there's another molecule I haven't mentioned called ANC5 that we will ignore for the time being. And what you see here is that binding of both tenurin and FLIRTs, we propose together by coincidence detection, activates latrophilins, inducing synapse formation. And so the hypothesis here is that latrophilins direct the specificity of synaptic connections by coincidence detection of two ligands in an isoform specific manner. Now, this would imply that latrophilins act as signaling molecules, as molecules that convey a signal for organizing synapses. As it turns out, GPCRs are signaling structures. They do signal, right? So that would imply that maybe latrophilins act as GPCRs. And in my final few slides, I will demonstrate to you that this is actually the case. To test this, we made a series of mutants, one mutant that has no transmembrane regions, so it can't signal, only a GPI anchor. Another mutant that has blocked G protein and arrestin signaling because it has an insertion in the last cytoplasmic loop, a T4L insertion, and thereby it blocks signaling. And a third mutant that has no cytoplasmic tail. These mutants fold well, they are transported well, so they can be used to test whether signaling is essential and whether it is mediated by G and protein arrestin binding using rescue experiments. And that's exactly what we did. And so we were focused actually on this mutant because it's the most specific. It's the one mutant that specifically blocks G protein arrestin binding. And we could show this in non-neuronal transfected HEC293 cells, as shown here, where we measure cyclic AMP levels and protein SK activity. I'm not going to go into this, just to demonstrate that when we have wild-type latrophilin 2 or wild-type latrophilin 3, they stimulate cyclic AMP and pKa. When we put a T4L in there, it's gone, demonstrating that they do have GPCR activity as a wild-type protein, but not after we actually mutate the G protein binding site and the arrestin binding sites. And so in my final slides, I will show you that this causes a loss of activity. If we look at latrophilin 2, as shown here, Schaffer collaterals are not affected by the deletion and not affected by the expression. Perforant path is affected by the deletion, rescued by wild type, but not by the T4L mutant. If we do the same thing for latrophilin 3, it's basically the same story, only now it's the Schaffer collateral that is impaired and the perforant path that's normal. So to summarize here, what I've tried to tell you in this part of my presentation is how we approach the function of a family of genes, latrophilins, that are linked to neuropsychiatric disorders and that we observed are functioning as synapse-inducing molecules, as signaling molecules that induce synapses, but induce synapses in specific dendritic domains and CR1 regions of the hippocampus. And thereby, this serves to address one bit of what I call the synapse enigma, which is our current lack of understanding of how synapses are actually built. This basically tells us how at least some of the synapses in the hippocampus are created in specific locations. And I want to end here I talked to you about two sections. The shorter sections are trying to tell you why it's important what we work on. A longer section illustrating an example of how we do this. I want to acknowledge the people because that's the most important part, the people who actually do the work. Rick Sando, Xian Zhang, and Shuchen Zhang did the work I discussed today, the more recent work. It was originally started by previous postdocs who are now in their own positions someplace else. And I'm grateful for the funding we receive for our work at Stanford. Thank you very much. I'm happy to answer any questions you might have.
Thank you very much, Prof. Südhof, for the excellent and very inspiring lecture. And very privileged to hear about your latest research finding in this session. So I've got plenty of questions streaming in, and there was a central interest in terms of what you have described in your disease modeling experimentation, in that animal disease model have not fully recapitulate a lot of the human disease biology, and that you mentioned that human genetic background is necessary to recapitulate this interplay of genetic and environmental contributor, right, to this overarching neuropsychiatric disorders. So would you enlighten us about whether there's a need to introduce any stress paradigm in vitro or in vivo, and in terms of even facilitating animal models as a preclinical model to do drug development, and how do we better recapitulate some of this human pathology in various disease models?
Yeah, that is an excellent question, because clearly the enthusiasm of animal models for human diseases has decreased, because in many cases human neurons, for example, but not only neurons, turn out to be different from the rodent ones. However, I still think that animal models are indispensable and that they're extremely useful. We just have to do everything with a degree of caution, because the human models are also not good. And let me tell you why. Okay, so organoids are pretty much the rage right now, but organoids never mature. They never really grow. And thank God, you don't want to have a real brain in a dish. They're great, probably better for non-brain than for brain applications, but they're basically flawed, fundamentally flawed. In culture, you can actually get more mature synapses than in organoids, but still, these are in culture. These are not in a real organism. However, with these real synapses in culture, you can at least look at human-specific features. Some argue that we should always go to primates. But you know, primates, there's a lot of them. And the ones that are reasonably accessible, they are so far from us evolutionarily, they're really not that close at all. They're quite distant, quite different. So I'm not sure how good of a model they really are. Moreover, I personally have some ethical concerns about using primates for everything. I think we should use primates as people. As humans, we should use primates as models only when it's ethically really necessary. So what I would argue for is a plausible and rational mix, a well-designed mix of different approaches that meet as best as we can the needs for drug development, for disease mechanism studies, and for mechanistic studies. But of course, it would be much nicer if we had one solution that fits all.
Thank you. So in your very recent studies on latrophilin, and the defects in latrophilin 1, 2, and 3, and how it has impact on the neural circuits in the hippocampus, why is it that there hasn't been much study about their impact on the cerebellum? Or is it a much difficult or hard to reach targets to study?
Published even a paper on the Latrophilins and cerebellum. You know, there's only so much you can do. I wish people would give me more money. And I wish more talented young people would want to work with us to explore these interesting questions. We're always looking for great people to do this. Yeah, cerebellum is great. The other area I would love to study is the prefrontal cortex, because that's so essential for all these psychiatric phenomena. But, you have to basically focus on the things first that are the most best suited for your question, and do as much as you can with the available resources.
Great. Yeah, that's a great advice. In fact, it's so important that we are designing fit for purpose assay to look at the impact of various mutation or risk variants that you have talked about. And in this latrophilin mutation defects is that when we try to relate something of a molecular underpinning to clinical manifestation, is that so some of the participants were interested that in neuropsychiatric disorders, where it's mainly defined as human behavioral perturbation. So how do we translate some of this impact on the molecular mechanism underlying latrophilin 123 mutation, and how that relates to the clinical manifestation?
Let me actually come back to the Neurexin 1 mutations, because there the situation is a little clearer. The observation that a heterozygous mutation, loss of function, causes a robust change in just one property of a neuron in a dish. Granted, it's only in a dish, but still, it's very, very selective. It's selective but robust. It's really interesting, I think, because it suggests that that mutation is actually biologically significant and tractable. At this point, there's a huge gap between the clinical observations of schizophrenia and other neuropsychiatric disorders and the genetics. For most genes that have been identified as being important for these diseases, there's no function. So you can't actually go anywhere with them, because there is no function, you don't know what to do. With Neurexin 1 in this case, we now have a physical correlate of the mutation, a functional correlate of the mutation that provides a lead, but only a lead. The obvious avenue would be to try to understand why that heterozygous mutation causes a decrease, and if there are avenues to correcting that loss by some other manipulation that then might lead to drug targets. The problem in neuropsychiatric disorders, and in fact in neurodegenerative disorders as well, is a lack of targets. We don't have any targets. We don't have any validated targets that really affect disease itself. We have only targets, we have only some drugs that are symptomatic, actually, for all of these diseases, but nothing that is disease modifying. If we want to go for disease modification, we need targets. If we want targets, we need more than genes. We actually need to know how it works. And so, yeah, it's quite a bit of work. There's no shortcuts, but I think there is hope, expectation that we will get targets and that these targets will be productive.
Great, thank you. I hope our young scientist participant will stand on the shoulder of giants like yourself to advance the field. And yeah, there's a hypothetical question about, do you believe that eventually we can connect the human brain to machines? And what do you see as the findings from biology transcending into areas like artificial intelligence?
Of course, we can connect the human brain to machines. We already can. And the way that works is because of the plasticity of our brain. We can train our brains to have certain electrical signals that a machine can read and then translate into some action. Yeah, it's great. I mean, there's nothing surprising about that because we know that we can actually train our brains. That makes it so wonderful. We can learn, and we can learn how to make our brains have certain electrical signals that the machine can read. So we can instruct the machine reading by training our brain. And these brain-machine interfaces, I think, will have a major effect, I hope, on the patients who have, for example, some paralytic event where they can't move certain parts of their body. I think that's terrific. But that doesn't actually change the fundamentals. It's a symptomatic help. Okay. Artificial intelligence is a totally different ballgame. Artificial intelligence is wonderful in many ways, but it's also limited. You need to train the artificial intelligence machines, if you want to call them that, in specific paradigms. They're much better at doing anything like pattern recognition than we ever will. A computer is 1,000 times better at memory than a human, 1,000,000 times better, a trillion, God knows how many times better. But that doesn't actually represent what I would call intelligence. The artificial intelligence is a different type of cognitive activity that is very, very helpful for some things, scary for other things, but doesn't really serve as far as I can tell, as a model for how the human brain works. The human brain works in many ways different. And that, I think, in some ways is good.
Sure. And I'm also mindful of the time, so I'm just going to distill just one or two more questions. That is, over here, widely recognized that cognitive behavioral therapy is effective in treating some of the neuropsychiatric disorders. And do you think there's interest to extend the study of synaptic functions in that how psychotherapy can impact on the processes at a molecular level?
I think these therapies are effective, yes. Because, that's part of the magic of our brains. They're plastic, they learn. We can train ourselves. And so what you do in cognitive and behavioral therapies is you're basically teaching the patient to better train her or himself to deal with their own problems. And I think that's wonderful. And of course it will change synapses. Synapses change all the time in the brain. And it will change synapses in a specific way. And I would love to know in which way, yeah. But cognitive behavioral therapies animal models are a little difficult here, yeah.
Sure. And for the interest of time and the interest of the participants who are mainly young scientists, that we know that through today, your inspiring lecture has resonated in a lot of us in that the decades of effort into basic science and addressing very fundamental biology is so important that later on allows you to facilitate those translational efforts to address what it means in disease. So would you have some last words of encouragement for scientists who are undergoing training in their pursuit of science in this current juncture?
Science is done for the joy of discovery, because it is truly rewarding to discover something. But the joy of discovery is not a purpose in its own. The joy of discovery has an even bigger meaning, I believe, for our societies, because discoveries underlie, in the end, all applications. There's a tremendous pressure in the United States, and I believe elsewhere, to go to applications directly and jump over the discovery, because there often is the impression that we know a lot. Well, as it turns out, we don't know a lot. And if you don't actually do the discovery, you can't do the application. Moreover, it turns out that the people who do discoveries are actually pretty good at doing translation. There's often the complaint that the scientist lives in his own little world and doesn't know how to actually do some translation. But I think that that's baloney. I think that's not true. I think all of us scientists who are really trying to solve scientific problems are eager to do applications. And I think many of us are the best suited to do the applications because we know the systems, we know where the opportunities are. So I think that in order to create a vibrant environment that basically produces new applications, produces new discoveries, new drugs, you also need an environment adjacent, close by, somewhere in sort of touching distance that basically is trying to do discoveries and not just applications. Because if you only want to do applications, I think you're going to fail. You're going to be a contract research organization. And so whoever is out there, I can't see anybody, whoever is a scientist, it's not just the joy of discovery. Whatever you discover, at some point in your life, probably your own life, it will actually allow you to do applications. You are doing a service to society that is essential for our society's survival.
Thank you very much. It's truly encouraging. And sincere thanks, participants sending their appreciation for you. They allow us to understand how we advance medical knowledge through basic science. Thank you very much, Prof. Südhof.
Nice to meet you. And it was an honor to give this lecture. Thank you.